Hi there, boys and girls. Welcome to tonight's vodcast on genetics and heredity. Now, genetics is the study of how traits are inherited through the interactions of structures called alleles, which we'll talk about in a little bit. However, heredity, the big idea behind genetics, is the passing of traits from parent to offspring. So here we have a celebrity couple. Brad Pitt's the dad, and Angelina Jolie is the mom. And here's their daughter, Shiloh Jolie Pitt, from a couple of years ago. If you take a look at their daughter, you'll notice that their daughter is a nice mix of characteristics from both parents. So if you take a look at their daughter's eye structure, she obviously gets it from her dad, Brad. If you take a look at the height of her forehead, she gets the height of her forehead more from her mom than she does her dad. If you take a look at her lips and her mouth area, you'll notice that that's a combination of both parents. The shape of the lips come from Brad Pitt, whereas the fullness of the lips comes from her mom. The reason why Shiloh Jolie Pitt looks like this is because she has inherited these characteristics from each parent. Her heredity is mixed because she inherited different alleles from her parents, which is the same reason why everyone else looks like a combination of their parents. So let's take a look at what alleles are. First of all, alleles are different forms or versions of a trait which are controlled by the genes. So at the same time, alleles are different forms or versions of a gene. So as we've seen this before in our DNA chapter, genes are the instructions to make proteins, and essentially they're just sections of DNA. So if we take a look at the upper right hand corner here in this dotted line box, this section of DNA is a particular gene. Now, as we stated before, genes have different versions and they are called alleles. As we know, genes and what they do are solely based on the orders of the adenine, guanine, thymines, and cytosines within that section of DNA. For example, here we can have a gene to make brown eye color based on the order of bases in this picture. However, if someone has blue eye color, the order of those adenines, guanines, thymines, and cytosines would be different. So the alleles are different because the base pairs are in different orders for the different alleles. So let's take a look at some examples of what alleles are. Now, as I said, different versions of genes are called alleles, so kind of like alleles are like ice cream. As we all know, there's a certain way to make ice cream, but differences in the recipes call for different flavors. As we take a look here, each one of these flavors, like the fish food and the chunky monkey and the chocolate fudge brownie and cherry Garcia, those could be alleles for ice cream. They are all ice cream, however, they're different versions of that same kind of ice cream. And the same goes for human characteristics, except we don't count ice cream as characteristics. We have different characteristics. So, the gene to make eye color. Everyone has a gene to make some sort of eye color, however, people do have different eye colors. Some people have the blue eye allele, which is the gene that makes blue eyes. Other people have brown eye allele, which is the gene to produce brown eyes. However, hair color can have different alleles too. So we all have genes to make hair color, however, not everyone has the same. So for example, there's an allele or a gene to make blonde hair, like Taylor Swift has. Or there's a gene to make brown hair, like third baseman for the New York Mets, David Wright has. So again, alleles are just these different versions to make these particular traits. We all have the ability to make these traits, but we just have different colors and different versions of them. So let's talk about how this is all discovered. So the idea of inheritance and the passing of traits from parent to offspring began with this man, Gregor Mendel. And he was the first one to follow traits through several generations of offspring and use mathematics and use probability to prove it. So what he liked to study was obviously plants because he was a gardener that worked in a monastery, and one of his favorite plants to study was a pea plant. And the pea plant was a good plant to study because it had many different traits to it, such as pea pod color, or seed shape, or plant height, or flower color, and so forth. So we can study all these different characteristics. Well, what we're going to talk about tonight is his experiment on pea pod color. So Gregor Mendel took two different colored pea pods. We had green pea pods and we have yellow pea pods. And then he pollinated them, which means he took the pollen from one of the pods and then fertilized the eggs of the other one. So, for example, we could say he took the pollen from the green pea pod and put it on the yellow pea pod flower to see what would happen. Now, most of us would probably think that if we took a green colored organism and mated it with a yellow colored organism, the offspring would be some sort of a mix between green and yellow. However, what Gregor Mendel found in his first generation, when he watched the pea pod develop from the first offspring group, he noticed that all the offspring were green. There was no yellow color. It was all green color. So he got to wondering, well, what happened to the green color? Was it destroyed? Did it go away forever? So what he ended up doing was he took two of those pea pods in the first generation and then crossed them and fertilized them. And in the second generation, he noticed that out of every 4 offspring, he would get 3 green pea pods and then 1 yellow pea pod. So let's take a look at how this works. Now, before we move on, there are certain phrases that you need to know in order to understand what's going on. When we talk about genetics, it's like learning a brand new language. So the first thing you need to know is what a dominant allele is. A dominant allele is a gene or an allele that covers another gene or an allele. So for example, the green color was the dominant allele in our example with the pea pods. And when we write out a dominant allele, we always use it as a capital letter. It's always written as a capital. So the green color would be expressed as a capital G. A recessive allele is a trait or the allele that gets covered by the dominant allele. So the yellow trait in this experiment was the recessive allele because it was covered in the first generation, but then showed back up in the second generation. And they're always written as lowercase letters. And if you notice, we just use one set of letters. We use capital G for green, and then we use lowercase g, not y, but lowercase g for yellow. And the other 2 words that you should know are purebreeds and hybrids. Purebreeds are organisms that have 2 of the same alleles. They're either 2 dominant alleles or 2 recessive alleles. Purebreeds are always written as either 2 capital letters or 2 lowercase letters. And then we have our hybrids. Hybrids are organisms that have 2 different alleles, one dominant and one recessive. So they're the opposite. As we write out a hybrid gene combination, we'll see a mix of a capital G and a lowercase g. If you think of a hybrid car, you'll know that a hybrid car has 2 different fuel sources. It has an electric fuel source, and after it reaches a certain speed, it'll kick into a gas fuel source. So, since it has 2 different fuel sources, it's called a hybrid. Well, here we have 2 different alleles, a dominant allele and a recessive allele, making a hybrid offspring. So let's take a closer look at his experiment here. Now, as we said in the first experiment, when Mendel crossed the 2 parents, there were 2 pure breed parents. So again, that means they were either 2 recessive genes or 2 dominant genes. Being that the yellow turned out to be recessive, we would write the yellow as lowercase g, lowercase g. And then the pure breed green, which turned out to be dominant, was written as capital G, capital G. Now we use this tool called a Punnett Square here, and the Punnett Square helps us figure out the probability of what types of traits are going to show up in the offspring. So Punnett Squares always have 4 squares in them, and each square represents an offspring that will be made. So when you set this up, what you do is this. You take a parent and you put their gene type or genotype at the top here. So since we have 2 boxes here, and I've put the green parent up here, I'm going to use capital G, capital G. So I put 1 capital G over the box, and then the other capital G over the other box. And then I took the yellow pea plant, and I put their genes next to certain boxes. So I have lowercase g here, and lowercase g there. So each box has a letter over it or next to it. Next, what we'll do is we're going to fill out the Punnett Square, and the Punnett Square is easy to fill out. All you have to do is just bring in the letters into each box. So for example, the dominant green allele I'm going to bring into this box, and then recessive yellow allele I'm going to bring in that box. Whenever you get a dominant allele and a recessive allele paired together, the dominant or capital is always written first. It's never the other way around. That's an incorrect way to write it. And we're going to fill in the Punnett Square the same exact way. Dominant allele green, recessive allele yellow. Dominant allele green, recessive allele yellow. And the dominant allele for green and recessive allele for yellow. This is where you need to understand the rules of dominance and recessives. Whenever you take a look at the genes in an offspring, if there's ever a dominant gene paired up with a recessive gene, the dominant gene is always going to show. So since in this cross or in this mating pair, the dominant gene is green, which is shown by the capital letter, this offspring is going to be green. And since every other offspring has a capital G in it for green, every offspring is going to be green. So as a result, Mendel found, as we said, that all of his plants were green. Whenever you cross two purebred plants together, you're going to get whatever the dominant trait is. So let's take a look and see what happens in the second generation to give us the results that we talked about earlier. So as we said, Mendel took two of the offspring from the first generation and crossed them, or mated them. So the eggs were fertilized, and each plant was a hybrid plant, as we saw in the previous cross. This plant is going to be big G, little g. This plant is going to be big G, little g. So we'll set up our Punnett squares just like we did before. One parent on the top, one parent on the side, and each letter over a box. And then now we're going to fill out the Punnett square. We'll drag down the capital G and capital G, bring over the capital G, and bring down the lowercase g, bring down the capital G, bring over the lowercase g, and in this last box, we're going to bring down our lowercase g and bring over our lowercase g. So if we go by our rules of dominance and recessives again, anything with a capital G, the dominant trait, is going to be shown. So green is dominant, so every pea pod that has a capital G will get a dominant color. So this has two dominant traits in it, so it's going to be green. This offspring here has one dominant trait in it, but still it's dominant, so it's going to show green, as well as this one. However, if we take a look at the fourth offspring, we'll notice that there is no dominant trait there. It's two recessive alleles. So the two recessive alleles will express themselves, and this pea pod will be yellow. So as a result, Mendel found that when he crossed two hybrids, he got three dominant plants, which were the green plants, and then one recessive plant, which was the yellow plant. So here's a summary of what Mendel discovered. So what Mendel discovered after his two crosses was this. Some traits, when present, cover up other traits, and those traits would be the dominant ones. So every offspring that has a dominant and recessive always showed the dominant color. However, the hidden traits or the recessive traits aren't gone forever. They're still present in all of the offspring here, except for this one, but they're not shown. But when you pair up the two recessive traits together, they then express that recessive trait. So that's how you fill out a Punnett square, boys and girls. And that's how the idea of alleles and heredity works. So what I'd like for you to do now is go into the back of your note packet and fill out the practice Punnett squares. Thank you for your time.